back to the last day of the session. I believe you all have enjoyed the whole 1 week. We actually enjoyed a lot interacting with all of you. And I assume that offline participants enjoyed yesterday dinner, because there was a kind of extended dinner with faculties and spouses, and we had a DG as well with his spouse. So that is a rare occasion. It's just that our workshop was happening. These two occasions were combined together. So it's rare for us to have a memento and certificates given to participants by Director General, normally Director. So it was a very good, I would say, sort of pre-closing ceremony yesterday. Today, as I mentioned day before yesterday, that we'll have a little extended timeline. So instead of 2, we will be closing at 3:30, because Dr. Ramalingam will explain to you a little bit of interesting online tutorial, so even online participants can learn and ask questions about his own expertise, which I believe is extremely useful for all of you. There's a lot of troubleshooting you can do with the kind of explanation or tutorial or a little bit of small exercise that we will take you through. So, just take the online participants also to see how Dr. Ramalingam explains that. And then the offline participants will go to the lab to see the whatever leftover work that is left, the HPLC analysis, discuss all the results that we have seen in the last 3 days. And we are going to come back here at around 3:20 because at 3:30 we have a closing remark. So I request all the online participants to remain here at 3:30, because we also have a Department of Biotechnology, a very senior representative coming to look at the closing ceremony or closing aspects of this workshop. And it will be useful because many of you young scientists who are looking forward for writing a grants proposal to Department of Biotechnology or any other government agencies, you can ask this question that you are asking me, that how will I write a successful grant so that DBT will fund. So hopefully she can't, because it's difficult for us to get time from the senior officials from DBT. So she is a coordinator of Bioenergy from DBT side and all the related areas, industrial biotechnologies and all that. So I request all the online participants also to come back and be here at 3:30, 5-10 minutes before is better. We are certainly going to be here in this auditorium at 3:20 approximately. So all of you, after the demonstration and after the discussion of all your 3 days of work, you come back here at around 3:20, and then you have a closing remarks. So with this, let me introduce today's speaker, Mr. Ramalingam. I hope there is no question. So Professor Ramalingam is a chemical engineer, a very good friend of mine. So he's a chemical engineer turning biotechnologist. He specializes in fermentation of genetically manipulated systems in bacteria. In fact, he has written bacteria, but he has also done a lot of fermentation in fungi, I know that. Dr. Ramalingam has a well-structured industrial career spanning 5 years in 3 giant pharmaceutical companies. So he's the person who has actually spent time in the industries, and those kind of questions if you have in your mind, scale up, commercialization, so he's the best person to talk about. So he's offering bioprocess and metabolic engineering courses for BTech and Tech PhD students at Anna University, Chennai. Earlier his group was working on the synthesis of green chemicals and biopolymers such as lactic acid, propionic acid, 1,3-propane-diol, hydroxypropionic acid, polyhydroxyalkyl, etc. Nowadays, he is actually focusing on cell-free metabolic engineering. So that's the aspect that is the focus of his current research in multi-enzyme in one-pot process and kinetic modeling of the pathways of interest that is used for in silico design for optical conversion. So novel small segmented pathways in vitro is chosen, the raw material having a low price and product consisting of many active pharmaceutical ingredients. His group is developing products such as L2-aminobutyrate, citron malate, methylacrylate, octobutanate, and beta-alanine. They're all very interesting industrial molecules that is being developed in his lab by different chemoanalytic processes. So with this brief introduction, I invite Dr. Ramanathan to give his lecture.
Good morning, one and all, both the online and the offline participants. And at the outset, I would like to thank Dr. Shams Saeed Esdani and Shirish Srivastava for providing me an opportunity to be a part of this prestigious event. And I thank Dr. Shams for giving me a nice introduction about me. And today I'm going to talk about the challenges in the heterologous pathway engineering, and more specifically my second lecture will be pertaining to determination of rate limiting steps in a biosynthetic pathway. So in my metabolic engineering, two aspects are very important. The first thing is that you need to make a committing metabolite in certain quantities as it requires. For example, if you want to work with green chemicals like ethanol or lactic acid or isopropyl, any of these green chemicals, when you want to compete with the chemical route, you need around 100 grams per liter. At a yield of greater than 90% with a volumetric productivity of 1 gram per liter per hour. These three things are absolutely essential. In order for that to happen, the first thing is that from a simple carbon metabolism, the cell should be able to commit that sufficient amount of that metabolite. If it is a pyruvate or acetyl-CoA, it needs to commit that. We need to make the cell to commit that. Once that amount of metabolite is committed, then any of these chemicals will have their own pathways. Probably from a main microbial chases, you can bifurcate to all these products, and it involves pathway engineering. So the control of gene expression is very crucial in such pathways, linear pathways, to convert the entire of the committed metabolite into forming the product. So my talk will be focusing on the second aspect, that is, once the committing metabolite is made in a certain quantity, how to convert them efficiently into product. And this concept holds good for any product, including biofuels. For example, yesterday we were discussing about butanol biosynthesis. So I have just put this figure here. So acetyl-CoA is a committing metabolite here, and from there on, you need a couple of enzymes, and which needs to efficiently convert acetyl-CoA to butanol. So there are so many other aspects, even in a linear pathway. The same enzyme may catalyze a few other reactions forming byproduct. The redox has to be balanced. So many other issues pertaining to metabolic engineering is a challenge, but nevertheless, we will focus on how to really maximize the conversion. And adding up to the previous theme which I was talking to you, when you want to make a cell to commit a certain amount of metabolite, in this case, as I have taken the example of succinate, phosphonol pyruvate is the committing metabolite. For the cell to commit enough amount of this phosphonol pyruvate, they have made a lot of knockouts, and they have shown that how by increasing the knockout numbers, they are able to increase the succinate productivity. So this particular aspect is primarily to do with systems biology approach, which Shirish might have given you flux balance analysis and op-knock tools and other things, whereby they'll tell you which are the targets have to be manipulated so that more amount of the metabolite will be committed. But then later, we need to focus on converting this phosphonol pyruvate efficiently to succinate fully. And what are the challenges involved in a multi-step pathway? One of the major problems in maximizing conversion is the regulatory hurdles. These pathways have evolved millions of years back, and for millions of years old is getting evolved, and they are very highly regulated. There will be feedback inhibition of the product, and also intermediates also keep inhibiting the enzymes. Allosteric inhibition will be there, and these regulatory hurdles have to be overcome. So once we started constructing a heterologous pathway, a synthetic pathway, then these regulatory hurdles have to be taken into consideration. The second major problem is that it will go for side reactions, toxicity of the intermediates. When the intermediate starts to accumulate, it leads to toxicity, and also it leaks out of the cells. And also quantifying the enzyme expression levels is important, and it gives burden. When you put huge amount of multiple enzymes to be cloned in a cell, then it creates expression, it creates burden, metabolic burden for the cell. So these are all the major challenges that needs to be addressed when you are constructing a heterologous synthetic pathway. So there are a lot of synthetic biology tools that have emerged whereby the expression can be controlled, but it is mostly, to my knowledge, is done by trial and error. So to convert that entire product into substrate into product, so we need to really figure out what is the amount of enzymes required and what is the ratio in which all these enzymes have to be synthesized. Thereby it leads to the entire substrate getting converted to product. So one of the best ways to go about doing this is a kinetic modeling approach. There are kinetic modeling approaches done at the genome scale. That's a completely different method. But kinetic modeling of a small segment of a pathway is what we are going to talk about today. And that can be done by using the actual enzyme kinetics by which each and every step proceeds. Since the number of steps in a pathway, individual pathway may be within 10 or 5 or 3, mainly 4 or 5, we can do this kind of an approach whereby each and every step we can find out what is the mechanism of action of that enzyme from the literature or even by performing experiments, and start developing the kinetic expressions for each step, and use these expressions to predict the trajectory of product formation and all the other, and also what are the most important thing is that there are so many regulatory hurdles, but what is the impact of each and every single regulatory hurdle on the flux? That is the key. There may be a regulatory hurdle which will cause a 10% lowering of the flux. I don't think you need to bother about it. Anything which is giving a major impediment to the flux flow, I think you need to address them in the first. And then you have to keep proceeding towards the regulatory hurdles, which gives a very low increase in flux. So for all these purposes, kinetic modeling is very essential, in the sense you can take an enzyme, develop all the basic kinetic parameters and fit it into the model. The model will take care of the rest. So thereby we can minimize a lot of trial and error experiments that will be required to fix the amount of enzymes required and also the ratio of the enzymes. So today I'm going to demonstrate this with the example of propionic acid. Okay, and this work was done in our lab. We published this in AMP journal, and the idea behind this work is that one of the best naturally metabolically engineered product is lactate. Okay, nature has designed this method, and an organism like E. coli or engineered E. coli or lactic acid bacteria can produce anywhere between 100 to 200 grams per liter of lactate at a yield of greater than 90%, at a volumetric product of 1 gram per liter per hour. That is the reason why this is one of the products which competes with the petrochemical route, and lactic acid for a very long time is being produced by biological method. And what we want to convert this lactic, such a high amount of this flux, into propionic acid, which is used as an animal feed. And also we want to, using this acrylate pathway, we want to produce this array of products. That's what is the basic objective of this endeavor. So we clone this acrylate pathway from Clostridium propionicum into this, into E. coli and lactic acid bacteria, which are supposed to be the best lactate producers. And we're supposed to get 100 grams. When the entire pyruvate gets converted to the entire lactate gets converted to pyruvate, we're supposed to get 100 grams per liter of the product, but we got 0.3 to 3 grams per liter. Upon optimization, we got 3 grams per liter. While the amount of propionic acid I required to commercialize is 100 grams per liter. Anything less than that will not be viable, commercially viable. But then we started to understand why it is not really getting converted. Why this 100 grams of lactate is not able to get converted to propionic. So that's our question. And in order to answer this question, we thought that we need to pull out all these enzymes of the acrylate pathway, assemble them in vitro, and start to study them and find out, hey, why is it not my cell able to convert this lactate into propionate efficiently? So we started to figure out that initially when the cell has to make the propionic acid from this acrylate pathway, it uses acetyl-CoA to supply the CoA to this pathway, and later on this propyl-CoA will start to set in the cyclic pathway. So initially for the first few seconds or minutes, it will be using acetyl-CoA. Once the pathway is set in motion, it will start and we need to validate the simulation, what we obtained from the modeling with the help of a cell-free system, which I have explained a little earlier. And it's a very interesting system for us because we need to break all the cells, express each and every enzyme independently, and either purify them and add them together and carry out the reaction, or else we can use the lysate and straight away use the lysate and carry out the reaction. Perhaps we work with the lysate because it mimics far more close to the intracellular milieu. So we need to validate our simulation with the help of these experiments, and we can also mimic the intracellular conditions by providing a buffer at the physiological pH, and for macromolecular crowding they advise some 9% PEG to be added, and for each and every organism we can formulate this medium so that it mimics the intracellular milieu much more closer than the simple buffer we use. And so we started to do that experiment and also the model predictions. Okay, the dotted lines are the model, and the bold lines are the experiments. And we found that model is able to mimic the experimental values. And what we found to our surprise is that this propyl-CoA got accumulated. So propyl-CoA got accumulated. That's the first observation we found. So in a pathway, so this propyl-CoA is accumulating. So which means that this step is the late limiting step. So that's what the model predicted, and experimentally we also tried to validate it. Yeah, yeah, yeah. It predicted, but I was not very interested. After doing the experiment only, I started focusing attention upon my student, and whatever he said, I started to believe. Until then, I was not very impressed. So this part of the work is published by us in Biotechnology, Bioenergy in the year 2020. So the same graphs I'm showing you. For different concentrations, we try to perform experiments. For certain concentrations, it didn't really match very well. Okay, but a few other concentrations it matched. Perhaps the model has to be refined. We have to work extensively upon the model and then get it refined and make them work better and better. And the important thing here is that this modeling, kinetic expression is it's not it's a mechanistic expressions. So once it is perfectly done, then the model will perfectly work. So that's the advantage here. So that's what we did with the cell lysate. We expressed in both E. coli and N-lactis. We used both the cell lysate. Both of them have got different set of parameters. So this is for the N-lactis I'm showing. The previous graph is for E. coli. We cloned it in E. coli. So, but nevertheless, the concept remains the same. So once we have this model in place, which is validated, it's fairly good. The next best thing is to we can use here we did the model itself, and we can also predict it by experiments. And this perturbation analysis is very important to fix the ratio of the enzymes. To fix the ratio of the enzymes. For example, this is the baseline level of enzyme concentration that is available, and it gave a propionate concentration of 246 millimole. Then you increase one enzyme by 10-fold, and you see how much does this increase, which means this step is rate limiting. So 0.02 to 2.2 to increase it is increasing because other two steps are kept constant. Again, you increase the other enzyme by 10-fold and see how much does the propionic acid productivity is increasing in terms of millimoles per liter of cytosol. So it jumps fairly high. So that means PCT enzyme is rate limiting for this concentration. Again, you change this LCD by 10-fold. So there again, the propionate productivity levels have increased. So which means that all these three enzymes are limiting to some extent. That's what we have observed. And further, you can increase this PCT by 10-fold in all these three experiments, and then keep increasing LCD. Okay, and here we have increased ACR again. But when you increase ACR, there is no increase in the productivity. So we can clearly see that ACR is not rate limited. This enzyme is not rate limited. And even if you increase it by another twofold, again there is no increase in the synthesis levels. So likewise, when you increase the PCT from 0.16 to 1.6 to 3.2, then also there is no major change. And also LCD, they doubled it. I don't think any major changes there. So from this, we can understand what should be the ratio in which these enzymes have to be expressed. No, this we did it with the model basically. Model, because we have the model with us, we did model. But the same experiments can also be, ideally we are supposed to do it with the experiments also and verify. Yeah. So what we observed is that we found that the propyl-CoA is inhibited. Enzyme I was telling you, propyl-CoA is getting accumulated because propyl-CoA transferase is inhibited. So propyl-CoA transferase is inhibited by lactate and the propionate. We found that. So because of the lactate inhibition, you need huge amount of enzyme to be put. So once that you're going to have an enzyme which is without inhibition, what will be the level of expression that will be required? So that is what we ascertained. So propyl-CoA transferase without inhibition means you require just 10-fold increment is more than enough. But if you are going to have that inhibition in that enzyme, then you have to have increase it by 80-fold. and all these enzymes have to be increased by this many fold. So that is the knowledge it gives us. Yeah, initially for first few seconds of it will be working, but this is not important. I think this is the important thing, because that actually proceeds. So what we found is that when you go to make the same enzymes from Clostridium propionicum, where this enzyme is having a limitation, having inhibition by lactate and propionate, then probably the amount of enzymes in terms of the percent of the total protein required is, imagine 47 plus 17 plus 2, okay, 67, close to, you have 70% of the total enzyme should be your enzymes in the cell. Only then you will be able to convert all that lactate into propionate. Then we thought that it's not going to work. It's impossible to express that much amount of enzymes, and we thought we can even stop the work because it doesn't lead anywhere. Then we started to think of constructing a heterologous pathway from multiple enzyme sources. And once we started, as the cyclic model predicted, the actual expression levels is what units per milligram of protein is what we got in E. coli and lactic acid bacteria. And in Clostridium propionicum it was 10 times more. And in our model it was 100 times more. Almost virtually you need 100 times more expression than what we could obtain in E. coli and lactic acid bacteria, simply by putting them in plasmids. We use P. coli and P2 vector, compatible dual vector systems to clone all these genes, and it looks virtually impossible to get that thing. And later on we started to scout for the same enzyme from other sources, and we found that there are enzymes which are far more superior to the enzymes in the Clostridium propionicum. And this Clostridium propionicum was provided by the person who worked for 25 years in Germany, and he provided us. He's the pioneer of the acrylate pathway. But we ended up having the worst enzyme. In fact, the product producing organism is not having a good set of enzymes. And the ones which don't produce, they all have very good enzymes. And we found we could found out that, for example, Ralstonia eutropha, they have propionic transferase, which has a very high specific activity. Okay, and we also found that they don't have inhibition to propionate and lactate. And similarly for lactyl-CoA deioratase, you can see the specific activity of Clostridium propionicum 61, while it is 1500 in that range. See the difference. Similarly for lactyl-CoA also, we have got twofold lactyl-CoA reductase also, we have got twofold increment, but this is not rate limiting step, that's what we understand. So this we can dramatically improve, and also this inhibition can be overcome by cloning these enzymes. And the same, so we also try to quantify the extent of rate limitation, which is what I am going to talk to you in the tutorial today. This number is very important. If it is somewhere around 0.5, 0.4, then this step is a major rate limiting step. As you can see, of the three enzymes, these two enzymes have a flux control coefficient. That value is around 0.5, and that indicates they are equally rate limiting, and they are the most rate limiting. And the ones, this lactyl-CoA reductase is having a very low flux control coefficient value, which means that they are not rate limiting. So we need to work only on the other two enzymes. And how with lactate concentration this flux control coefficient changes, that's what we have plotted here. And initially, before doing this analysis, we thought ACR is the rate limiting for various reasons, but we found it is actually not rate limiting at all. That's what we found. And so we did the modeling exercise, and then we fed these kinetic parameters of these enzymes into the model. We found out the expression level required is around 15% of 14 plus 3, yeah, 16% of the total protein. So from 70% now the amount of enzymes required had been reduced to 15% of the total proteins, and it seems to be a possibility. So that's where it is. And, yeah, so once these enzyme expression levels are ascertained by this modeling exercise and validation by cell-free system, then we can implement synthetic biology tool sets to control the expression levels to precisely produce the required amount of enzymes so that we can completely convert the lactate into propionate. And what we found is that why don't we use the same cell-free system to produce products? Earlier we were using the cell-free system to just understand how the regulatory hurdles works in a pathway and how to prevent the intermediates from getting accumulated. Later on we thought, why can't we use the same cell-free system to produce products of commercial value? So we started to focus on producing API, active pharmaceutical ingredients, drug intermediates by the cell-free system. And yeah, so currently we are using cell lysine because we are working on amino acids which can easily be crystallized by providing the isoelectric pH and by adding solvent. So it has a very powerful purification strategy, so we are working with cell lysine. Otherwise, it's preferable to work with immobilized enzyme. Purification is expensive. Unless until the product is very expensive, we can't go for purified systems. One of the products which we are working is alpha aminobutyrate. Alpha aminobutyrate, and it's a key drug intermediate for making levetiracetam, ethambutol, and briviracetam. And it is currently manufactured by China. They are the global leaders, and we are importing that entire amount of at least majority of it is imported. For example, Reddy's lab has a chemical method, but it's not the best like the Chinese method. And it is one of the 53 key drug intermediates, levetiracetam. And currently the product cost is between $17 to $20 per kg. And the global demand is 4,000 metric tons per annum. India produces 1,000 metric tons per annum. And the market demand is $1 billion in 2022. And the major utilizes US, EU, Japan, India, and China. This is how it gets distributed. And in India, the best produces are lupin, and we are in association with Aurobindo Pharma and Divis Laboratory. They are the major producers of levetiracetam, and so therefore they are the major utilizers of this intermediate. So the current process in China, they make it from threonine, and it's a two-step pathway with one step for redox balancing. So they use formate dehydrogenase. And the cost of threonine, even though it is available at $1 per kg for animal feed, they are not able to use it. So Aurobindo, they said they can't use anything less than $3 per kg threonine. So that level of purity is required for them to make this intermediate. And we developed this process from citraconate. And this is isoleucine biosynthesis pathway used by very few bacterial species. And it comes from citramalate, from pyruvate through citramalate, it flows into this pathway. And this goes to make the isoleucine. And here we wanted to just substitute some other enzyme which is able to catalyze 2-oxobutyrate to L-terminobutyric acid. So this citraconate can be obtained from citric acid by a single step vapor phase catalytic conversion at a high yield, and it is quoted at $2 per kg by AB Enterprises at the scale of 6 tons. We're going to buy 6 tons per month, they'll be able to provide at this cost. And when we're going to make it, we are sure the industry is acceptable that we can make it at $1 per kg. And this is also a 3-step process like like the ones here. It is redox balanced. This step NADH is formed, and here NADH is consumed. Here also ammonium formate. It is also costing $1 to $1.4. So totally 3 plus 4, 4 to 5 or $5 to $6. It is costing to have the process. Substrate cost itself is $4 to $5 in this process, while in our case it's only $1. So that's the major difference that it creates in the process advantage. The second major advantage we have between this process and that process, that all these three enzymes have very high catalytic efficiency, kcat, into the range of 10 to the power 5, while here also it's fine, but loosened dehydrogenase is not, is 10 times or 100 times less than balanced dehydrogenase, which we are using. So, and also formate dehydrogenase is not a very good, very efficient enzyme. Though a lot of enzymes are involved, its kcat is never close to 10 to the power 5. So that way our enzyme requirements also are less. So these two things are major advantages in this particular process. And this work is filed in the national phase filing across the various regions, USA, Europe, Japan, China, and India by Bioenergy. And they also filed one more patent for this valid dehydrogenase, which are mutated to increase the activity by twofold. So we are working on increasing the expression level of all these three enzymes. As you know, except this first enzyme, one Luby enzyme, the second enzyme, the pathway, all the both of the enzymes, they form inclusion bodies. And we use heat shock, sorbitol addition, low temperature to have solid expression. And right now, we were initially planning to have around 10 units per milligram. Even 1 unit per milligram is good enough. That's how we started conceiving. But now we are able to get around 70 units, 20 and 50 units per milligram. So the amount of enzymes we have produced is tens of times more than what we really envisaged. So thereby we have the flexibility of increasing the enzyme concentration. And last year we got this 50 grams per liter. The industry, ideally, the cost of L2-aminobutyric acid production will decrease anywhere from $3 to $6 when you go between 50 to 100 grams per liter. But they wanted 50 grams per liter to start with. And we found that after some time it stops. So then we need to figure out why it's stopped. We have lots of problems we solved. And once, even after reaching the 50 grams, we begin to see that it's stalling. And that's mainly due to polyphosphate. That's what we realized. And we solved that problem now. And as you can see, the curve is increasing. This is the dream line I was looking for all these years, where I want a single straight line where it keeps going, and I should be able to stop where I want. So right now we are able to achieve around the molecular weight of alpha aminobutyrate is around 100. So we can say that 500 is 50 grams roughly, and 60 grams. And earlier we got 75 grams per liter. That's at 36 hours. We got in 36 hours. Right now you can see that we are able to do it in probably this is 200 minutes, more than 3.5 hours, and now it is 180, less than 180 minutes. So 2.5 hours we are getting. So 3.5 hours, 2.5 hours we are doing it. So we want to keep it within 5 hours. We want to keep it within 5 hours, and so that the equipment size will get reduced. The product time gets reduced, the size of the equipment get reduced. The size of the equipment get reduced, fixed cost will go down, and that will indirectly impact the operating cost. Yeah, 39 degrees. I'm sorry that I don't want to reveal most of the data, so I'll just put some numbers. It's a temperature. So we are finally also working on the temperature. No, conversion done at 39 degrees. Yeah, this is done at 37 degrees. One is in a batch process, other by fed batch process. This is at 39 degrees. Yeah, in vitro. Yeah, we add the enzymes, and we vary the enzymes. 4 enzymes we are using. Yeah, 3 for the step and 1 for providing anaerobic condition. we need anaerobic condition for the first enzyme, loose CD. It is having iron sulfur cluster. It requires reduced environment and anaerobic environment. We can sparge nitrogen on a large reactor, but it will be expensive. So since we have NADH with us, we added NADH oxidase. Very small amount of 0.5 units of NADH oxidase we are adding, so that it will scavenge all the oxygen, so that you do not sparge nitrogen. So you will probably have to aminobutyrate. Yeah. Yeah. Yeah, with 2 millimolar, 1 millimolar, 0.5 millimolar, even less than we need ideally. Yeah, recycling. So industry does biotransformation, but they use, they move groups. They want to hydrolyze, they want to isomerize. So they don't work on pathway enzymes as such. But that is very much similar to this cell-free metabolic engineering. So they have broad specific enzymes which they have to mutate it or screen thousands of enzymes which will work effectively for the particular substrate. But here in metabolic engineering, we take enzymes from the metabolic pathways and they are evolved by nature. So there is no need for engineering. So that is a major advantage. Yeah, the oxygen availability will be very low in the reactor. So 10 milligrams or less than that. Even if it's going to scavenge, then we are working at 100 grams per liter. So this reaction is 100 times, 1,000 times more than that reaction, even if it proceeds. So small amount of energy is used, it's not going to be a problem. Yeah, see, this major challenge with this cell-free metabolic system is that, for example, in our system itself, for example, the third enzyme, malin dehydrogenase, it requires manganese to replace ions. Okay? And this requires Mn2 plus, and this inhibits lubin. Okay? Likewise, if you have conflicting, then we settle for a compromise. This is very good at pH 9. These two enzymes are very good at pH 7.5. We are working at pH of 8. Okay, so we make compromises, but that compromise should not really impede the process in a very big way, and it should not make the enzyme to be added a very huge amount. And stability of the enzymes is very important, and so many factors to it. So, but the important, interesting thing is that all these decisions can be made before starting the work. So that's the major advantage. So it's not that there's no exploratory component. For a successful technology development, these things are very important. One is that it should be scientifically very simple, and techno-commercially it should be very powerful, and it should have least exploratory components in the process. Everything else should be visible. Like your idea is novel, but every other aspect should be available to you. And finally, it should be easily executable. These things, both things are hallmark of technology development. And so we have so industry wanted this 50 grams per liter, but 60 grams per liter or 50 or 60, but we wanted to go anywhere between 80 to 100 grams per liter. That's what we are looking at. And once we reach that, then our cost of manufacturing, in 5 to 6 hours we have designed this process economics, and it comes to around $3.5. If it is 50 grams, probably it will be around $6, $5 to $4, $4 to $6 it will be there. And we work on all of these substrates. Even buffer, for example, working with goods buffer, heap-based buffer is 6,000 rupees per kg. Tris buffer is 1,000 to 1,500 rupees. It's impossible to use those buffers because it will have a huge fraction. So we are working on triethanol mine, and you can see it is costing only $1 per kg, 70 to 100 rupees per kg. So we work on every aspect we need to look into so that the cost can be kept under control. And it is $3 per kg. The product cost is $18 per kg, and then industries will be willing to take it. If you have even 1 unit of enzyme per ml, 24 hours we can get around 150 kilograms per meter cube. But we are putting huge amount of enzyme now because we are getting the whole process in 2.4 hours. That's where we are looking at. And the second. No, we are producing these enzymes. The cost of manufacture, I'm not showing it. The enzyme cost also we are calculating. Yeah, how much it costs to make that enzyme. E. coli enzyme. Yeah. No, we are not purifying. We are doing only ultrafiltration. Only ultrafiltration. Because Arabindo and all, they want to take it to the next step even without purification. They say the product should have 95% purity. If it is there without purification, they can go to the next step. Because making Limitras citrum is 4 steps away from this step. So it is not even the key starting material. So without purification, they want to shift to the next step. Because purification will cost some money, even crystallization. Sorry. Yeah, the second product we are working with is beta-alanine. This beta-alanine is a health supplement, and it's used for bodybuilding for the fitness purpose. And it is also can be this beta-alanine is 3 aminopropionic acid. It can also be combined with pantolactone and cysteamine to make pantotene. It's a cholesterol lowering health supplement, and it has got a huge market value. A couple of industries are manufacturing it. And also pantothenic acid, vitamin B5 also has a huge market. 10,000 tons per annum is the global market. And it costs anywhere between $10 to $35. This cost very high, probably to go down. And pantolactone is cheap, and isobutyrolate is also cheap. So the costliest compound is only this. And it costs anywhere between $8 to $10 per kg. And we want to produce this beta-alanine from this 1,3-diaminopropane. And 1,3-diaminopropane can be obtained from 1,3-dichloropropane by a single-step ammonolysis. And this dichloropropane is a waste product, and it costs $0.1 to $0.2 per kg. So, which means that the diaminopropane should be made at $1 per kg, and then the cost of making this should be $2 perhaps in this case, because the product cost is not very high. And it should also be attached with all these steps, and so that the same industry can make the final products. Then the process becomes viable. In ADH oxidase also we produce it in the E. coli, we put it inside. While we are making these enzymes, 3 enzymes, we will integrate that gene into the chromosome perhaps. A very small amount of the NADH oxidase will be allowed to produce later on, so that we don't put any extra effort. So it will come along with those 3 enzymes and go into the process. Yeah. We purified things will be expensive. We don't require such a purity also. At no cost we will be making it. So this process has 2 steps process. One uses diamine oxidase, the other one is 3-aminopropionic dehydrogenase, and thereby we are able to convert beta-alanine. And this is the initial stages of the process. And we are waiting for the funds, and the hydrogen peroxide generated will be recycled to oxygen by catalase. Here it will be recycled by NADH oxidase again, and water forming NADH oxidase we have to do. But all said and done, we require 0.5 moles of oxygen for every mole of conversion. So in this process, we may have to we are providing aeration, and but still these enzymes work. So that's how it is. And they are my students, past and present. And our photograph. And earlier they were my collaborators for the work which I described in the beginning. My Indian collaborators and my institute and IIT Madras and few of the collaborators from abroad. And for the cell-free system, I am doing it alone. Nobody to join me. Probably if I succeed, many more scientists may, because I already succeeded now. I've got this required commercial viability. It has to be scaled up. And I'm a scalar person, but sadly I don't have the facility to scale it up. I don't have the money to scale it up. So that's the problem. And, but then I think we will surely scale it up. That will not be an issue. And the important part of the scale up is scale down. So scale down means whatever that happens in large scale, that should be mimicked in the small scale. For example, you are sterilizing a medium. It takes 40 minutes to heat up and 6 hours or 7 hours to cool down. So if you're going to do that in your reactor in the small scale, you have to take 40 minutes to heat it, allow it to cool for 6 hours, and then the medium composition will exactly mimic the same medium composition in large scale. So this concept is called scale down. So once your scale down is proper, then scale up will never be a problem. So scale down and scale up are very crucial. So always try to mimic the conditions at large scale. And imagine that anything you do cannot be extrapolated to the large scale. So those are the things which we need to look at. And thank you for your attention.
Interesting. I didn't know about very successful cell-free system that you established.
Yeah.
I always used to think that only 2 grams per liter would be a limited principle. 70 grams, 60 grams per liter of transformation efficiencies. I think the key is that the balancing of the redox, right? And the pathway you've chosen is very intelligent pathway because they are self-balancing.
Yeah, I think we are working for years on that. So naturally, enzymatic biotransformation are not able to do this work because they have never used this pathway. We are constantly, for Shams, for example, all through his life, his mind will be filled only with pathways. He may not be a good biochemist also, I'm not sure about that. Ilayaraja is not knowing the Carnatic music, but he can combine those ragas into new tunes. So likewise, he is a master of combination. All the time he combines. But you will be the people who can appreciate this work the most because you struggle so much to do the manipulation inside the cell. We struggled for 15 years. My students struggled, and that's how I thought, hey, how should I get over this problem? And how to get over people with the expertise of professors like him. That's what I thought, and finally I got this idea. And initially I saw some papers in very high impact journals like Nature and all. Even a few milligrams were reported. So that was very discouraging for me. But some other work I saw.
I also saw some of the papers. I didn't go into the details, but they do suggest a very good cell-free system for large populations. I haven't seen the details, but there are some success.
Yeah.
The thing is, if you would implement the knowledge of cell-free system to the in vivo system, then how do you manipulate or ensure the particular ratio that you are able to generate in vitro? For example, let's say 47 is to 16 is to 2, or whatever the ratio of the 3 enzymes that you are able to generate. So generating those kinds of proportion in vivo is going to be a difficult.
No, in vitro also it is not successful in the sense. But that's what we need to go for heterologous pathway, and the expression level should be less than percentage of 15%. Beyond that, we'll have metabolic. So we cannot produce product to 100 grams per liter like recombinant protein production. The cell dies after some time. No, it starts with lyse. But in metabolic engineering, we can never afford to do that. So our expression levels, in my opinion, cannot exceed 10% of the total protein, 10 to 15%. So we need to. Yeah. Now with a lot of screening mechanism tools, both bioinformatics, in silico and otherwise, we can scout very efficient enzymes which are not used by the organism. That's the interesting thing. The producers don't have the very best enzymes, while the non-producers have the very best enzymes with them. So that is, I think, is the key.
Let's ask question. Go ahead, please ask questions. Anything that is coming up in your mind for this presentation.
Is there using emulsifications to put cell-free systems in and then use.
If they are going to use lipase kind of thing where solvent is there,
Yes.
then these problems may crop.
Okay.
Yeah, I've never worked with interface, so I want to keep systems very simple. So I've never worked with that such a problem. Yeah, that can be a problem.
Thank you, sir, for a very exciting talk. I just want to know what are the major advantages that you see in these cell-free systems over expressing those same enzymes, those same heterologous pathways in E. coli or some other organisms. What is the major advantage that you see?
One is
And
that
what conditions you would like to do those pathways in cell-free systems?
yeah. See, inside the cell, you can have a huge pathway, correct, from glucose to any of this product. But cell-free system, you cannot have that. Number one. Number two, you can never manufacture product of low value, say ethanol or biofuel, any other biofuel, any green chemical. You cannot do it by cell-free system because cost of manufacturing itself is costing $3, $5. The product has to be expensive. It has to be $10, $20. And these are major limitations. And not always, you will always find a metabolite which is very cheap, and after 3 steps, it is going to be 100 times expensive. So we constantly, my mind keeps searching that. So every metabolite price, or how can I synthesize a metabolite very easily from a chemical step? That chemical should be a rock bottom price. From that, I need to make this substrate very cheaply, and from there, I have to make the product. So lots.
You can try to express these enzymes in E. coli, and you find it difficult to do that. Then you move to the cell-free system.
Yeah, initially in the alpha-aminobutyric, it's a collaborative work between University of Georgia. We made citramalate. From citramalate, we wanted to make this alpha-aminobutyric. So we could clone those genes into the E. coli, and we initially wanted to make 2-oxybutyrate. We got few milligrams rather, very less amount, and we approached the industry. Once we designed to produce this product, I approached the best Lupin, for example, largest producer of this product, and they said metabolic engineering we don't want, because in India the power cost is very expensive when compared to other countries. So fermentation is very expensive. So it requires chilled water, cooling water, aeration, and agitator power, steam. They're all energy intensive. So, but cell-free system, the reactor will be very large, the agitation will be very slow speed. So the operating cost will be 1/5 or 1/10 of the fermentation cost. So investment cost is also very low, because the amount of enzymes we require will be for 1000 meter cube reactor. The enzymes will be required will be 100 liters or less. So now 1000 meter cube means 10 times or 100 times less. So
But for expressing those enzymes, you have to do fermentation.
that's what that fermenter size will be 1/20th the size of the conversion vessel. If I'm going to have 1 meter cube reactor, the enzyme fermentation vessel will be 100 liters or 50 liters, 30 liters. So all put together will be 100 liters. So the size of the fermenter becomes smaller, everything becomes smaller. Compressor facility, chilled water facility, cooling water facility. So investment cost drastically goes down. So pharmaceutical companies, they want enzymatic biotransformation. And they don't even prefer wholesale biotransformation. Then we thought we can go for wholesale biotransformation. They said, again you have to go E. coli on a large scale. Then again the same problem. So we don't want wholesale biotransformation also. So they want very efficient enzyme. So you should need to produce them in a very small fermentation facility and put them into a very large reactor and carry out the conversion. That is what the pharmaceutical companies want. So that's why we shifted to that. And there are a lot of limitations in cell-free system. But I, as an academician, I have the flexibility to choose whichever is the best I can showcase. That's why I've chosen these two products.
Thank you, sir.
No, we are not currently purifying because both these amino acids at isolated pH, they add solvent, it get crystallized. They can also recrystallize it. So they don't prefer purification.
You just lyse the bacterial cells and you lyse it.
Yeah. Currently we are using ultrafiltration to get rid of all the small molecules, phosphates, and also we are using alumina to scavenge the polyphosphate. But later on we want to go for gene knockout to prevent the cell from accumulating polymers. That's what.
Thank you, sir.
Hello sir, my name is Lokesh. Thank you for this interesting talk. So from your presentation, I see that you have made a kinetic model, right?
Yeah.
Genome scale kinetic model.
No, not
So-
genome scale kinetic model. A segmented pathway kinetic model.
Okay, so basically in stoichiometric modeling, we do flux balance analysis, and one of the that is basically optimization. So in that, one of the constraints we use is steady state mass balance. But in a kinetic model, you will be integrating the reaction kinetics into the equations. So your equations will be a function of time.
Time.
So you cannot use the conventional steady flux balance analysis, which we do it at steady state mass balance. So
Yeah.
my question is, what technique had you used to make these simulations, and what is the principle behind it?
How do we do this modeling?
No, the simulation. Your yes.
No, we feed these kinetic equations into, we are using Coppasi.
Okay.
It's open software, you can anybody can use it. So we feed these kinetic equations into this model, and you can give different substrate concentrations, and all the trajectories will be created by the model.
Okay. Coppasi.
Substrate, yeah,
Okay.
substrate product, intermediates, all these things. Steady state analysis also will be done by that.
Okay. Thank you.
You can create a steady state system also.
Oh.
Yeah, you can continuously keep adding the enzymes, removing it. You can maintain the constant enzyme concentrations in a container, and you can measure the product, substrate, intermediates at steady state. All this possible.
Thank you.
The model also does that.
Mhm. Okay.
So at the larger scale, how do you envision your system as a CSTR sort of thing, or I mean a continuous system or a batch?
This is a batch only for
Right
synthesis.
now you have, but I'm talking about when you are taking to the next step.
Only when you're going to have an immobilized system. For example, if it is an immobilized packed system, packed bed reactor kind of a system, then it can be operated continuously.
Correct.
For a high fructose corn syrup, a lot of other processes they do it like that, a continuous process. Because this to drain the reactor itself will take 1 hour, to load the condenser will take 1.5 hours, the process will be only for 2 hours. That means only 50% of the reactor time is effectively used.
Right.
The remaining 50% goes waste. But if it's going to be a continuous process, then the reactor size will come half or even less than that.
Correct.
So, yeah, that's the preferred one.
Good morning, sir. My name is Sandeep Kumar. I'm working on a scale up product with Dr. Srivastava. I have one little question to you. As you are increasing the size of the reactor for the enzyme or whatever you are producing E. coli, so what is the cost is coming for the E. coli recombinant enzyme?
What is the cost of recombinant enzymes? $150 it is costing. If I'm going to use 1 meter cube reactor
Okay.
of fermentation, it is costing anywhere between $100 to $150 to produce whatever is the enzyme level and all. And we're going to have a cell density of around say 90 OD, that is 30 grams per liter of biomass. Then
So-
it costs $100 to $150.
So you are producing these enzymes in your laboratory using fermentation or in C class?
We have reactors. We have reactors of 2 to 5 liters capacity, 20 to 300 liter reactor. And we produce both in the shake flask and the reactor.
Okay, and then same enzyme you are using for the
Conversion.
conversions.
We use Ependogs, some 10 ml falcons, round bottom flask and reactor. All these for the conversion. We don't take lot of trials. We need small volume. Once we are sure about that, we keep juggling in different volumes.
Thank you. One more thing I like in your presentation, as you have worked with the industry and you are working in research, every step you have calculated the cost of the process because you want to transfer the thing. As we are working on bioenergy, so suppose nowadays petrol is $100 using a lower vehicle thing.
Yeah.
So that's why I like your talk too much. Every time of point you are calculating the cost.
Thank you.
It will be.
Yeah.
Thank you.
We are very conscious. My students are trained to be very conscious that way. So that's the only way we ensure that the cost doesn't overshoot. They never add more enzymes. I tell many times you add it and show me at least. It's okay, but they never do that because they say even if it works, it's of no use because it overshoots the. Yeah.
Any other question?
Continuously from one side to get the product.
Yeah, we do it with batch, but batch sometimes have substrate inhibition is there, then we do fed batch. We feed little by little.
And
Sometimes
then those enzymes, how much duration they are active in cell-free system?
enzymes, enzymes have to be stable. Our enzymes are stable for 24 or even 48 hours were stable. That we ensured in the beginning. If it is not stable, then you have to stabilize it by immobilization. And
So like you are making cell-free system means you must be doing cloning first, then you lysate it,
yeah,
and then those are expressing and doing the reactions.
yes,
So you have to go through the cloning process.
yeah. The advantage of the cell-free system is that you can use any expression system. One protein can come from E. coli, the other can come from Pichia, the third one comes from fungus or animal cells, doesn't matter. So that's another advantage of the cell-free system. While that is not possible with our metabolic engineering work. No, yeah,
If you use three different systems, then you won't have to worry about level of expression.
yeah. In vitro that's not a problem. Only in vivo that is a problem. In vitro we can add, if it is viable, we can add any amount of enzymes.
Right now you are using different systems or same.
We are working only with E. coli. Because the industry prefers E. coli. Somehow they don't even prefer Pichia. You know, they say we want only E. coli. Yeah, but like I said, that organism's natural enzyme producing ability is very low. See, these are pathway enzymes. They are secretory enzymes, they produce it for making substrate and other things, substrate utilization. This will be very small amount, so we need to overexpress it. Yeah.
The inclusion bodies, then how do you take care of that?
Yeah, so we developed a method of heat shock, sorbitol addition, low temperature. For
So
all
what is
the
heat
protein,
shocks? Heat
But-
shock.
yeah.
What is this heat shock?
no, we raise it to 47 degrees. For 15 minutes again we cool it
Just
Okay.
before induction.
before induction. So sorbitol addition we add 50 gram per liter and low temperature 20 degrees. We found that almost 99% of the protein gets solubilized in these three conditions.
And sir, this the lower temperature after induction for how long you are keeping?
16 hours.
16 hours.
Initially, to start with, we don't struggle with the inclusion body. So we have a method whereby any protein will be solubilized. To some extent, we can work on it. Then we started to optimize it.
Okay, thank you, sir.
And also, fusion proteins can be used in these purposes. Maltose binding protein, for example, high amount will be there, soluble expression will get. Cell-free systems, we have to go for it. We are also working on it.
Any other question? Any question from online participants? You can just unmute and ask question. I think it was mine. If anybody wants to ask question from online, just unmute yourself and speak. I think we need online participants also to actively participate in asking questions. Okay, maybe during the tutorial it will be more interactive. Just an announcement for online participants, so your certificates will be sent via email. And in case you want your certificate Okay, so we are going to take break and come back at 11 o'clock in this, we'll come back around 11 o'clock, and then Dr. Ramanam will give you some tutorial and online kind of analysis of some of the questions that are being asked, that how to make the, you know, enzyme proportion and kinetic models and whatever you want to just teach them.
Only basics about what is the flux control coefficient. That's it's an application of flux analysis.
It will be good to learn about it. So we'll come back at around 11, and then start with Ramanandam's tutorial, and then maybe after 45 minutes or 1 hour we will go to the lab, and at that time I'll share you with the video of today's demonstration as well. Okay, so let's break for before just a minute, I need to one moment. also here, so I will request you.
So I'll try to do exercises to understand how to estimate rate limiting steps in a linear pathway. So that's what we will try to do. First, understand the basic concept and how to really determine it. It can also be done by using modeling. Yeah. Can you hear? So these exercises and the concepts are taken from these books, Metabolic Engineering by Stephanopoulos and Introduction to Metabolic Control Analysis by Salto. And also many of the slides are taken from Dr. Dirk Martens of Bioenergy University. I went for teaching an exchange program in metabolic engineering, so I got some slides. Would use the slides as well. Okay. So today the tutorial is about metabolic control analysis. So far you might be working on metabolic flux analysis, and here we're going to learn about what exactly is metabolic control analysis. When you have a pathway, a linear pathway again, and this entire metabolic control analysis is done at steady state, just like how flux analysis is done at steady state, this control analysis is also done at steady state, because in steady state that all those differential terms will vanish. They want to have linear equations. Everywhere they want to have linear equations. In order to have linear equations to make life simple, people use steady state. Here also is a steady state. And earlier in biochemistry books, they have indicated that the first step in a pathway is rate limiting. It's a non-irreversible reaction. There will be feedback inhibition kind of a thing. But very often you find that not so. And at steady state, one can never say which is rate limiting. All steps you, for example, a grandmother and a granddaughter are walking at the same speed on the road. An observer standing on the side can never find out who is actually rate limiting, because both of them are walking at the same speed. You may presume that the grandmother is old, so she's walking slowly, because of the granddaughter is also walking slowly. But in fact, she may have been injured in her leg, and because she's injured, she's walking slowly. To match her pace, the grandmother is also walking slowly. So which means at steady state, you can never say which step is rate limiting, because all of them, you have a pathway with 5 enzymes and 4 intermediates. All of them work at the same rate at steady state. So these metabolic engineers, so they came out with certain methods to figure out what exactly is rate limitation. Does every step contribute to rate limitation? How to quantify rate limitation? How to quantify rate limitation? Once you do that, then you can precisely say, hey, these are the steps which I have to work upon. Whether I need to work upon the entire pathway or whether I can work on few steps, that's the first decision we need to make. If you need to work on all these enzymes, that's a very difficult task. But if you have very few enzymes which needs to be worked upon, or those enzymatic step is a major rate limiting, then we can easily manipulate them. So for that reason, this understanding this rate limiting step is very crucial. And it's a central goal of metabolic engineering. And this is a hydraulic container, and where we can have an analogy for this enzymatic pathway. For example, the amount of flow that is in the container can be compared to the flux in the pathway, and the amount of water in the container, each and every container, can be analogous to the intermediate levels maintained at steady state. They are different levels, but they are maintained. And the walls, will indicate, the size of the walls indicate the enzymes, different enzymes having different catalytic efficiency. And the amount of opening can indicate the amount of enzymes that work. And the important thing is that if you open this wall a little more, then this level will change, this level, all these levels will change, flow rate will change. To what extent it changes, that's very important. Which means that if you make a perturbation any aspect in this entire pathway, it will be felt across all the enzymatic steps in the pathway, almost in all the pathway. So that's what it's called a global global parameter, global variable rather. So here we can see that V3 is said to be the, not V3 perhaps, V4 is the major rate limiting step. So how do they quantify a rate limiting step? That's the first question we need to ask. So what they do, scientists they try to change any one enzyme in a pathway. For example, you take one enzyme, this particular enzyme, you need to change that enzyme by a very small amount. With a small amount you increase the enzyme and see how much, because when you increase the enzyme by a small amount, in that particular step the velocity will increase. That small increment in velocity, how much it can contribute to increase in the flux in the whole pathway. Flux I mean by the velocity in the pathway, entire pathway. Okay? Flux is velocity in the entire pathway. If it means velocity in a single pathway. Single enzymatic step. Okay? There are distinction between the two. So that's the slope. Delta J by delta E. Now this bracket is for the concentration, enzyme concentration. So this is important. This ratio gives you the measure of the control that the enzyme has on the flux in the pathway. And that is not the same for all the enzyme concentrations in the pathway. So it is in relation to all the other enzyme concentrations in the pathway, and therefore until a certain point this will very much affect the flux, but the enzyme concentration goes beyond certain level, it will become very excessive in relation to other enzymes, and virtually it will have no control over the pathway. It will hardly change. So the flux in the pathway also keeps changing with enzyme concentration. That is because it is in relation to all the enzymes of the other steps. So if I'm going to take this ratio, and if you want to measure this in millimoles and or moles, this value will keep changing. So in order for this value not to change based on the units of measurement of this, we're going to take the relative change in the flux or the relative change in the enzyme concentrations. So what you're going to do, you're going to take a small difference in the A value, measure it 2 different points, find the delta E, and take the point at which the concentration of the enzyme is determined. So same way the flux also. So delta J by J, delta E by A, because the units will get cancelled, and that's the major advantage. And this is called the flux control coefficient. And this flux control coefficient, in order to do this method, if it is a fungus, for example, you know cloning tools available, then probably they do classical methods of breeding homozygotes and heterozygotes, whereby they can bring about the differences in the expression levels. But as of now, the best method is to go for increasing the gene dosage by using plasmid or integrating the gene to the chromosome. So by such methods, we can alter the enzyme levels. And before altering the enzyme levels, you have to measure the flux in that particular pathway. And flux you have to measure by flux analysis, by metabolic flux analysis, or by flux balance analysis, what he has taught you. All of them will give you fluxes in the pathway. Then you have to alter this enzyme, any one enzyme which you are interested in. And again you measure the flux in the same pathway and see how much difference that flux has come about for a certain level of enzyme change done. So that is what is important here. So flux is usually measured in terms of millimoles or carbon millimoles per gram of dry weight of your cell per hour. So that's the quantity. So J is that, and enzyme activity is the enzyme concentration we are talking about. So one of the drawbacks with this estimating method is that you need two values at a very close points. Then only you can find out the delta J. When you change the enzyme, there are very low differences there. You will have a lot of random errors because you measure it. So, the first major problem is that you cannot vary the enzyme level by a very small margin. It's very tough to do that. Even if you're able to do that, measuring that small change in the flux will have a lot of errors. So we have to do a lot of experiments and draw a best fitting curve. So scientists were puzzled as to how to really solve this problem. Then they came out with a large deviation method. This method is fairly easy to implement, experimentally. So in this method, what they did is that most of the curve for the flux change with respect to enzyme is hyperbolic. Okay, it's a rectangular hyperbola. So they presume that it's like Michaelis Menten kinetics, that the equation half saturation kinetics. So this curve represents that. So once you have that assumption, then even if the J value is you're taking at two different points, still you can calculate the flux control coefficient. That's what it is. And how do they do that? Okay. This doesn't go there, huh?
Yeah, it looks like it is not.
Yeah. Is it clear now? Okay. Yeah. I'll stay put. Yeah. So basically, it's there are different methods by which we can estimate this flux control coefficient, and this flux control coefficient is normally done by modulation of the enzyme content by using large deviation method, and we can also reduce the enzyme activity by using inhibitor. So I'm not going to touch upon this because it's much similar, and they also use this flux control coefficient from elasticities and connectivity. We'll talk about what is elasticity and connectivity. Here, what happens is that measuring flux is a little tough, so that if you have one value, then you can estimate the flux control coefficient for any other enzyme concentration by using these elasticities and connectivity. It's far more easier. We'll see how we can do that. And also we can calculate this flux control coefficient from model. From Coppasi, software is there which we can do metabolic control analysis straight away. These are the methods by which, but they use the same concept. The models use the same concept. And that is how the flux control coefficient is estimated. And because we have normalized it's hanging. It's hanging. So this flux control coefficient, since we have normalized it, it takes the value of between 0 and 1. Flux control coefficient will take the value of 0 and 1. And that indicates that the sum of this flux control coefficients of all the enzymes in the pathway will be equal to 1. Okay? Which means if one enzymatic step has a control coefficient of 1, that means it is the one enzyme which is completely controlling the flux, while all the other enzymes are in excess, or rather they have no control over the pathway. Or if two enzymes are controlling, like what we have seen earlier, 0.5 and 0.5, that means two enzymes are equally controlling the rates in the pathway. So the value of the flux control coefficient will tell you to what extent is controlling the rate. The higher the value, higher is their ability to control the flux. Sum total is 1. So which means that if this rate limitation is overcome by putting more enzymes, then this step may become rate limiting. So the flux control coefficient will change. If you change one enzyme, the flux control coefficient for all the enzymes will change after that. It's not fixed. It's not relative. In relation to this step, if you increase that, what will happen? That's how it is. So whatever we measured in a cell, in relation to that we have expressed the flux control coefficient. But if I increase one enzyme by say by 10-fold, then these two flux control coefficients will change. Okay? So So that keeps changing. In a traffic, you have one major bottleneck at one point. If you remove that, then the traffic will get stuck at the next bottleneck, right? So there will be more traffic at that point. So if you remove that, it will go to the next point. It's shifting. It is shifting and dynamic. So there is nothing called this step is the major limiting step, or that step is the major limiting step. They are all in relation to all the other enzymatic steps. Okay? So all the relative enzyme concentrations inside the cell that decide which enzyme is limiting to what extent. But once you change it, then the whole picture completely changes, and each and every enzyme assumes a new set of flux control coefficient values. So that's the most important thing we need to do. Yeah, so what is this large deviation method? As I told you, they assume that the flux is always changed like a Michaelis method kinetics. That is, the flux will change with enzyme concentration like A, E by B plus E. This A is equal to V max in the Michaelis method kinetics, which is the maximum value of it. As A becomes larger and larger, this will become higher and higher. And the curvature, the nature of the curve, if it is very steep, this B value will change. How steep it is, it becomes smaller and smaller, the curvature becomes much more steeper. As it becomes larger and larger, it will become more shallow. That's how it is. And the interesting thing here is that if you know the value of A and B, that means for any enzyme concentration, you can find out the flux straight away. And by using this logic, they're able to show that at different enzyme concentrations, you can calculate the flux control coefficient straight away by using this equation. So here, as I told you, in large deviation method, the enzyme levels can be very much different. So okay. So if you take one point here for enzyme concentration E1, where the flux is J1, and you increase the enzyme maybe by 3-fold or 4-fold or 5-fold, and then you have a different flux value J2 at the enzyme concentration E2. We have 2 points, and by using these 2 points to calculate the flux control coefficient, you need to know the value of B and the enzyme concentration. And this value of B is calculated from this equation. And this equation they have arrived at by differentiating this equation, Tj by dE, and multiplying it by E by J, which is flux control coefficient. They got this expression. Okay? So don't worry about all that. This expression they got like that. And they equated these two terms with A on one side, and then they got this expression. But I think if you want it, you're interested, I think it is at the back of the lecture, which you will get it later, the derivation part. But the important thing here, what you need to know is that if you can, B can be calculated just by this expression where you need to know the value of J1 and J2, E1 and E2. If you have that value, then you can calculate B. Once B is calculated, then you can calculate the flux control coefficient for any enzyme concentrations. So all you need to do is that take a E. coli, grow them in continuous cultivation, measure the flux at one point, measure that enzyme activity. You express the same enzyme by cloning, or you change the inducer concentration, measure the increased enzyme level, and then do a chemostat, measure the flux again. Then from that you can straight away calculate the flux control coefficient. So that's how it is. That's what. A is the parameter. Both A and B are constant for this curve. They tell you the nature of how the curve will be. A will tell you how large it can go up, and B will tell you how steep it is. It can be shallow or it can be steep. It should be standing here. Is it shallow or yeah, no, not no, that's no. Enzyme, yeah, but here, no, here, how much does this enzyme control the flux at low concentration and at high concentration? That will decide the nature of the curve. probably not. That is, here, this enzyme, how it is able to change the flux, it is in relation to all the other enzymes in the pathway, not only this enzyme, independently. Yeah, yeah, yeah. It is in relation to all that. Just like I'm richer than someone. If I see a person who is poorer than me, I'm rich. If I see somebody who is richer than me, I become poor. So it's in relation to that. So the thing is fixed here. But this point, you're able to, because in a class I'll derive all that, the logic behind it, but I'm not doing it here, but straight away I'm going it. But all you need to know that it can be done by you. Simply for getting this expression, you have to, I can show that. Let's spend so much time, I can show this straight away. Yeah, we define this flux control coefficient is dj by j divided by d by EA. That is dj by j multiplied by EA by j. That's what it is. So we have differentiated this equation and we got this term. And after that we multiplied by E, and for j we use this expression again. We put it, and then we simplified it. Finally we got this. That's how we got it. Yeah, A is not required here. Yeah, A is not. Yeah. Interior lines. That is called elasticity coefficient. But here we cannot do it. Yeah. Instant rate of reaction of that particular enzyme. That separately, you have to pull it out and do it, that is different. That is elasticity coefficient. Yeah, that is what? That is flux control coefficient. That's flux actually. Sir has taught you, no? Metabolic flux analysis. 13 CMFA, metabolic flux analysis, flux balance. They all give you the velocity in the pathway. Velocity in a whole pathway is called as flux. That because chemical engineers did this. Mass get transported, heat does get transported, momentum get transported, they call it as flux. Anything that gets transported, they call it as flux. Here metabolite is flowing, so they want to call it as a flux. Whole pathway. Yeah. No, we are measuring only the velocity in that pathway. No, in a steady state, the velocity will be same in all the steps. All the steps. It's like water flowing in a pipe. You can measure at any point, but the velocity is going to remain the same. Because steady state, it's a steady state. Yeah, in a linear pathway at steady state. In a chemostat, when you're going to do an experiment in a chemostat, then all the velocities at steady state, all the velocities will, in all the pathways will remain constant. So that's how, that is the value of G. Yeah, so we will, now we can calculate by a small method. They derived it here. So, for calculating this flux control coefficient, you need this J1, J2, and this is divided by J2. So, here also E2. But in the case of small changes, it is E1 and J1. That's the difference. But anyway, we can calculate by both methods. Here I have told you, in small changes, okay, here I am. In small changes, you are going to get delta J by J value at this point, delta E and the E value at this point. In large deviation method, you are going to measure this, but J2 and E, you are dividing by J2 and E2. But in small changes, you will be dividing at the same point, J1 and E1. That doesn't matter. But there are two methods by which we can calculate this flux control coefficient. So what we will do, I have taken the values of E value Okay, don't worry about the units and J value. The same graph is shown in Excel, and I want you to calculate the flux control coefficient at 0.3, 0.5, 1 and perhaps 2. For these 4 points you can calculate. For 0.5 you can calculate for 0.5, 1 and 2. For these 3 points you can calculate by small deviation and also by large deviation. Can we do that exercise now? Yeah, 0.5, 1 and 2, because I have the values here. So 1 and 2. Yeah. You can take 0.5 and 1 for large deviation. For small deviation, you have to calculate the very next value, either one above or one below. At 0.5. Okay. Yeah. Yes. Correct. You have any difficulty? This value is there. You take one point above or one below. Okay? The change that is known as delta J. Yeah. And divided by the J value, 0.3. Okay? This is the enzyme. So this is the enzyme. Delta E by E. This is flux. You can take one value above or below. That is delta J. You divide by that value, that is J. Delta J by J divided by delta E by E. calculate both for small deviation and large deviation and see at that point how it changes. Yeah. Yeah, either one above or below. Yeah. 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 Fine. Yes. Difference, huge difference. Still it holds good at low concentration. 0.29, 0.4, 0.07, 0.12. They're almost only at higher concentration it drastically changes. Lower values of enzyme concentration it is fairly okay. Both small deviation and large deviation. Yeah, you can try that. We have seen how more and more large it is, it will be more and more erroneous. Very large deviation also, the difference becomes larger and larger, it can be more erroneous. Yeah. Yeah. Okay. That's okay. Range is how much are you getting? Point? Okay. That's okay. See, you are saying this only, no? It's closer to what I've calculated. Yeah. Shams is at 1. At 1 for large deviation stuff. Are the online participants doing it? Will you know that? Will you track it? You have any difficulty? Ladies and gentlemen in the online, should I offer any assistance?
Just ask any question, online participants, are you able to understand, are you able to calculate this? I think based on the equation that he has already shared before. So basically using the equation of B and
Yeah.
C that he has shared before and is asking to calculate the flux control. If they are able to, they are saying they are able to calculate.
Okay.
So if there's any questions, you can just ask.
Yeah.
The main thing is if you are able to follow the equation that we have shown before, I think first you have to calculate E and then you calculate C, then you
Yeah. 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 Yes. So, yeah. Yeah, what? It is the most accurate, but we cannot do it practically. So we need to rely only on large deviation. And large deviation seems to follow very closely to small deviation at lower values. Okay, lower values. At higher values, at higher values of enzyme concentration, it is very much different. from this exercise is that at lower values of enzyme concentration, the flux is more, right? Flux control coefficient is more, 0.29. And as you go at higher concentration of enzyme, the flux control coefficient keeps decreasing. That is what the graph is showing. What, 0.5? Yeah. No, it will be different. Slope is continuously decreasing. It will be little more, but not very much. Maximum, yeah. If you have 1 means it's completely controlling the entire rate. All the other enzymes have virtually no control. No, for one, in a particular enzymatic pathway, we picked out only one enzyme. No, we need to do this exercise for all the enzymes. Yeah, every trial should have only have to change one enzyme concentration little bit. And next enzyme again. That's what cell-free system is better. Because all of them will have a pathway. You are going to have definitely going to have clones in that. Because we're trying to integrate it into the genome and all. It's better to have these enzymes and yeah. Yes. This curve will come. Yeah. No, you have Arabidose induction, no? That will be much more superior, no? Yeah. We'll definitely get the change in enzyme for the levels, and you can ascertain it. That will be the simplest. But in simple batch filtration also, we can mimic steady state. Logarithmic growth phase, if you take the values, that is a steady state. Flux can be calculated. Yeah, all of you could manage to do that. Got the values close to what you have seen? Yeah. I've told you that the flux value is flux control coefficient is very valuable and important to increase the flux. And to what extent will the flux change depending upon the flux control coefficient? And that is what we need to learn. That's the most important part of metabolic control analysis. And for that, what they have done, they wanted to increase the enzyme by a certain ratio, by R, say 2-fold or 5-fold. So they want to increase E2 by a certain ratio. And then the flux is going to change by a certain ratio F. So in order to get that, they are going to divide this equation by J1. This will become J2 by J1. This will become 1. This will become R and this will become 1. And then they rearrange this equation to get F. That is what is, how many fold will the flux change if I change that enzyme by a factor of R. This expression they got it, okay, upon simplification. And here, interestingly, if the R value is very large, then this 1 can be neglected. R by R can be removed. So F will become 1 by 1 minus CGE, which means that it clearly tells you, we know that flux control coefficient takes value between 0 and 1. As this value becomes more and more, the difference will become smaller and smaller. Then denominator becomes smaller, this F value will be more, which means the flux control coefficient is more and more, then the increase in flux that is brought about in the pathway will be more and more. That is what is this graph saying. So, in order to understand this, you solve this problem. That is, let us assume that a flux control coefficient for an enzyme is 0.5 under certain conditions. By genetic engineering techniques, the quantity of enzyme or the maximal activity is increased 5-fold. Now all the other parameters of the system under study do not change. How much does the flux change by this modification? Okay, so now the previous equation, this problem says that they have given the flux control coefficient CJEA, they have given the R value, that is pi, and they are asking for F value. That's what they are asking for. So you can use this expression, take this expression. Yeah, this is 0.5. 5 times. Good. Yeah. Any difficulty? Yeah. How much you are getting? 1 point? 1.66 I got. Just check it up. Shall we move? Yeah. This is the simplification. What we understand from this problem is that when the flux control coefficient is 0.5, when we increase the enzyme by 5-fold, we got only 1.66-fold increase in the flux. Less than 2-fold only has increased. So this graph tells you for any particular flux coefficient, okay, so let's say 0.6. If at 0.6, if I'm going to increase the gene copy enzyme by 2-fold, then the flux changes by very small level. Okay, if I'm going to increase it by 5-fold, then the flux changes by say by 2-fold. Okay, if I'm going to increase it by 10-fold, then the flux is going to increase by 2.5 times or something like that. Which means as when the flux control coefficient is very low, even if you increase it by 50-fold, the flux hardly changes. Okay, only when the flux control coefficient is above 0.6, 0.7 or so, then if you increase the enzyme by certain fold, at least the flux changes by few fold. So which means that for any gene manipulation, your flux control coefficient be above 0.5. Below, if it is below 0.5, most important thing we need to find. Yeah, even upon 50-fold increase, you hardly see any change in the flux. So if you're going to have 5 steps in a pathway, all of them having 0.2, and it can't be like that. If it is equally distributed like that, then which means your whole pathway you have to improve. There's no way you can work on one enzyme. The entire pathway you have to alter. But of late, these concepts came long back. But now we can virtually do that. The synthetic biology, synthetic biology, we can do that. So, but still, it puts a lot of strain on the system, on the host, if we keep increasing the enzyme activity. So, the last part of it, here I'm not solving any problems. For the want of time, I removed it. I'm not sure if we have time. So the advantage with, we can indirectly calculate this flux control value, because every time measuring a flux is tough. You have to either do a batch cultivation or a chemostat, do the flux balance analysis, then calculate the flux. So we can indirectly calculate this. When you have one flux value, we can indirectly calculate by using other methods. So that is what I'm going to teach you, how we can make those measurements much easier. So one of the most important things that helps us to do that is by using elasticity coefficient. Okay, elasticity coefficient is very much similar to Michaelis Menten equation, where we try to calculate if you change the substrate concentration for that enzyme, how much does the velocity changes? Okay, you have an enzyme, you can pull the enzyme separately. You have one substrate, certain substrate concentration, you have some velocity. If you want to change the substrate concentration, how does the velocity changes? The significance is that in a pathway, at steady state, intermediate will have a certain fixed concentration, which I have showed you in the beginning with the hydraulic container. All the intermediate concentration will be fixed at certain value. If the value is going to be shifted by a small level, how much change in the velocity will be brought about in that step? And this analysis can be done in isolation from the rest of the steps. It can be done in isolation. You can pull out the enzyme from the pathway. Separately, you can have estimate that. So that's the advantage. And therefore, they assume that all the other steps in the pathway is constant. That's why they are using the partial differential. Now all the other flux control coefficient and all, every step will change, so you cannot trace them. But here, you can pull out the enzyme and easily you can estimate the elasticity coefficient. Okay? How? By changing the substrate concentration, the elasticity changes. The reaction velocity changes. The ratio gives the elasticity coefficient. And if inhibitor is there, it will have negative value. Okay? And even a certain portion of the substrate, it may have negative value. You can have both negative and positive values. And we can also use, as I've told you, the summation theorem. The flux control coefficient of all the enzymes in the pathway will be equal to 1. So once you have these two theorems in place, or even a few other theorems they have developed, you can use them as constraints or equations to solve for flux control coefficient. And they have another theorem called connectivity theorem, which says that you have a metabolite, single step, where substrate gets converted to this metabolite, and this metabolite converts into product. There are two enzymes catalyzing the reaction, and this, let us call this velocity V1, V2. And we can have flux control coefficient for this enzyme, and we can have flux control coefficient for this enzyme. Let the velocity in the whole pathway be J, that's what is flux. So the product of the flux control coefficient for this step and the elasticity coefficient of this step plus this two, flux control coefficient for this step and the elasticity coefficient of this step is equal to 0. So this is what they call it as connectivity theorem. And there's proof for it. I'm not able to show you here. And if you modify this equation, you can see that they are inversely proportional. When the flux control coefficient is large, then elasticity coefficient will be low, and vice versa. So by using this connectivity theorem and the summation theorem, for example, they are able to calculate the flux control coefficient only by using elasticities. Okay, by calculating these two elasticities, they are able to calculate the flux control coefficient for the entire part. And this is how Coppasi calculates it. Coppasi model calculates it only by this method. So this is relatively easy. You can pull out the enzyme, calculate elasticity coefficient for whatever enzyme concentration intermediate that is available in the pathway. Then you can perform this calculation problem. With this, I'll end up the tutorial part, and I haven't prepared for this kinetic model properly. I have got the Coppasi models of my work and then shown you how they have done it. And, but you can see the tutorials. You can go to the web and see through the tutorials. And I will also show it for a few seconds here. Open it and show it to them.
I think we should explore a few solutions.
Grammarly suggestions catch when your tone might undermine your
You'll have that species A, B, and C will be kept fixed. So when we start Coppasi, we get the main window with the logo. Because we're going to define the model, we need to go straight into the model part here, first entry, and once we highlight model, we get a page for the main properties of the model, starting with the name. Let's call it just branched. Below we have units of time, volume, and quantity. I like to change the volume unit to be liter, so I know everything is a multiple of liter and milliliter.
So we can go through the thing. Okay, drop down menu will be there. My biochemical model, and we have to determine the species. What are the metabolites that is participating? How many compartments? We are mostly working with the vacuum system, one compartment, and how many? It cannot work like that. Like model. Yeah, the substrates, products, everything you have to feed. That's what species is all about. In the reactions, okay, and that's what all the three reactions are there. They are having three reactions, and what kind of reactions they are. Okay, so they have a set of reactions like reversible Hill 1 modifier, mixer integrated non-competitive inhibition. So whatever enzymatic mechanism is there, you can choose. Or you yourself can feed the kinetic equations into this model. You yourself can do it. And the model values, whatever parameter values is there, we can give those parameter values to the model. And also we can make the model to calculate parameters, okay, by fitting. But that is not very accurate. It's preferable that we measure most of the parameters or if you have reliable data from the literature, then we can feed all those values to the model. Yeah, so this is how it will appear. Whatever formula, whatever equations they are feeding, it will appear like this. Okay. Non-competitive division, second order is here, irreversible. Then the discounting expression will be there. So for each and every step, we have to choose those reactions as I've stated earlier. That's how the model is created. Okay, then the model can be simulated.
Now,
So results will, plots will also
put
be there.
assistant button, find plot type, appear in plots, and creating
All time
appear the
course
next time,
analysis, steady state analysis,
other things.
everything will be, we can visualize it.
Let's
Plots,
close this plot
whatever
window.
plots you want, it will be able to.
Now we'll look at some settings which will affect the output to plots and tables. This is will not be apparent. Obviously, our duration should be set higher than this if we want to see anything.
I'll show you the steady state tutorial. So I'm not sure this Jacobian reduced system is not for our metabolic control analysis. I will probably go with another type of metabolic control analysis. This I'll keep the volume so that you can listen to it for 8 minutes.
Metabolic control analysis, MCA in short, is a mathematical framework used to assess how the control upon the steady-state values of the system's variables is distributed among the different reactions. Let's break this down. Let's consider this simple biochemical network. If we let the system follow its dynamics, it will reach a steady state. These two plots, for example, show how the concentration of the different species and the fluxes of the different reactions change over time. As you can see, after an initial transient, all the variables reach a plateau, and they don't change anymore. This means that the steady state is reached. Now, the question is, what happens to this steady state if we change the activity of an enzyme, for example, the enzyme catalyzing the first reaction? In general, we expect that the system will adjust to this perturbation and that a new steady state will be reached. This is what happens in our example, where at time 80 seconds we changed the activity of the enzyme. In particular, we doubled it. Now, we may want to quantify how different these two steady states are.
Now, where is another voice coming from? No, initially it was okay only. Did I make any changes now to myself?
More specifically, we may be interested in how much the steady-state concentration of a given species or the steady-state flux of a given reaction changed as a consequence of that perturbation. The control coefficients are used precisely for this purpose. There are two kinds of control coefficients in MCA: the flux control coefficients and the concentration control coefficients. The flux control coefficients are defined as the ratio between the relative change in the steady-state flux carried by some reaction and the relative change in the activity of the enzyme we have perturbed. Likewise, the concentration control coefficient is defined as the ratio between the relative change in the steady-state concentration of a species and the relative change in the activity of the enzyme perturbed. In both cases, the change in the enzyme activity must be understood as the perturbation we introduced in the system, while the change in the variable, either flux or concentration, is a consequence of such perturbation. The higher the control coefficient, the higher the control that the enzyme exerts on that variable. The reason for the perturbation to be taken infinitesimal is that the system's behavior is nonlinear. What we mean by that is that the response of the system, for example the change in the flux J or the concentration S, is not proportional to the change in VI. The values of these two ratios, then, depend on the amplitude of the perturbation. This introduces an ambiguity in how to define the control coefficients. To do away with such ambiguity, the control coefficients are defined for infinitesimal perturbations. When the perturbations are infinitesimal or very small, the behavior of the system can be approximated by linear response, and these ratios become unequivocally defined. Now, let's perform a metabolic control analysis in Coppasi. For this example, I use a modified version of Pritchard and Kell's model of yeast glycolysis. You can download the original model from JWS Online or Biomodels. To calculate the control coefficients in Coppasi, we select Metabolic Control Analysis from the task sub-tree and click Run. This is the result of our analysis. This first tab contains the values of the elasticities. We'll go back to this later. For now, let's skip it and go to the second tab. In this tab, we have all the flux control coefficients organized in a matrix. Rows and columns have the same name, but they mean different things. The rows represent the fluxes of the reactions, while the columns represent the enzymes that catalyze the reactions. So this, for example, is the control that the enzyme HK, hexokinase, exerts on the flux of the reaction catalyzed by TPI, triose phosphate isomerase. It's easy to see that if our biochemical network has n reactions, this matrix will have n by n entries. Please note that the control coefficients can be negative. This means that if we increase the activity of this enzyme, the flux of this reaction will decrease. Like in all matrices displayed by Coppasi, there is also a color code to help you. Intense shades of green are associated with higher control coefficients. Intense shades of red with highly negative coefficients. The pale cells are associated with coefficients close to 0. You will also notice that some entries of this matrix have the same number across a given column. This means that the control exerted by the enzyme on the corresponding fluxes is the same. The reason is that these fluxes are bound to be equal at steady state due to the stoichiometry of the network. As a consequence, a perturbation on the activity of an enzyme will have the same effect on the steady-state flux of such reactions. In this other tab, you have the matrix of the concentration control coefficients. The columns, again, represent the perturbed enzymes, but the rows, this time, represent the species that were affected. So, if we have n reactions and m species, this will be an m by n matrix. Again, the color code will help you to identify the highest coefficients and their sign. At the top right corner of each of these tabs, there is a drop-down menu from which you can choose between scaled and unscaled. The scaled control coefficients are those we have introduced before, and they are defined as the ratio between the relative change in the variable, either a flux or a concentration, and the relative change in the parameter, the enzyme activity. Sometimes, however, we are interested in the absolute change of these quantities, and this is when we use the unscaled control coefficients. The unscaled control coefficients are defined as the ratio between the absolute change in the variable and the absolute change in the parameter. Now, let's go back to the tab of elasticities. We consider them last because they are not usually the primal focus of modelers. However, they are important to derive the value of the control coefficients. The elasticities quantify the effect of a metabolite on the flux of a reaction if that reaction is considered in isolation from the rest of the network. In particular, they are defined as the ratio of the relative change of a reaction flux and the relative change of a metabolite participating in that reaction, either as a reactant, a product, or a modifier. In this case, the change in the metabolite concentration is to be understood as the causing event, and the change in the reaction flux as the observed effect. But remember that this effect is without the presence of the rest of the reactions in the network. So, how the elasticities come into the picture? In Coppasi, the control coefficients are not determined by actually perturbing the enzyme activity and recalculating the steady state. Only the reference steady state is retrieved, while the control coefficients are calculated through the stoichiometry matrix and the elasticities using simple matrix algebra. This makes the calculation much faster than explicitly perturbing the system and rerunning a steady state analysis after each perturbation. This is the main reason why elasticities are important in MCA, because they allow us to calculate the control coefficients by analytical means. If you are interested in the maths used to calculate the control coefficients from the stoichiometry matrix and the Jacobian, you can refer to these two papers. We hope you found this video tutorial useful. Thank you for using Coppasi.
Hello, in this tutorial I'll show how to configure and output text reports from Coppasi. The report can be custom structured for use in further analysis or visualization. Let's expand.
So if you have the kinetic model in place, then working with Coppasi is far more easier. For calculating two values, kinetic model, validating the model, change, for example, you are dividing it by J1 and D1. Okay? But in large deviation, you are dividing it by J2 and D2. That is what he was asking. Why it is like that or how? No? So J1, you are measuring at J1, but you are dividing it by J2 and D2. Or J1.
Any more question?
Learning the basic math is very important. Then you can do an operation by do that.
Okay, thank you, Dr. Lingan, for a very nice tutorial. Tutorial helps to learn. The rest of the thing we